All right, welcome to Chapter 6. When you look under the folder for Chapter 6, and obviously you've already done that, you're going to see three different sets of PowerPoint slides. There's a set A, B, and C. I'm only going to lecture over A. You probably should look through B and C, but it's not something I'm going to spend a whole lot of time, or really I'm not going to add some test material in there from those. That has more to do with specific muscle groups and labeling the names of muscles and those types of things. I might make passive mention of a couple of those, but I'm not going to get real caught up in that because I really want you to understand more about how the muscles work and how they create energy and do those types of things. So that's what we're going to focus on for Chapter 6, and we'll get started. This is going to be another one of those lectures that's going to be, there's a lot of stuff in here, and it can be very confusing. So I do have some videos in here, and you'll notice there's some videos underneath the folder section for this chapter. Please watch the videos. Hopefully between me talking about it and then you watching the videos, you'll have a much greater understanding. But don't hesitate to reach out and look for some other resources that's going to help you understand this a little bit better. Remember what I've been telling you guys all along, that learning is an active process. It's not something you can do passively. So you can't just sit and watch this video and expect to know this stuff. So be prepared to have to look it up and spend a little bit more time doing a little research, okay? So we'll get started, all right? So obviously what we're going to talk about today is the muscular system, and we're going to talk specifically about muscle cells and how those cells work. What you do need to understand that there are three different types of muscles commonly found in the body. Of course, skeletal muscles, cardiac muscles, and smooth muscles are the three that we're going to find. Skeletal muscles and smooth muscles are elongated cells, and they have what we call a muscle fiber. We call those muscle cells, we call them muscle fibers, so those two words kind of go hand in hand. The contraction of these cells causes a shortening of those muscles, and that's due to the microfilaments, the filaments deep within those cells that interact with each other in such a way that it causes that cell to get shorter. So it's a long cell, like a string. Think of it like the length of a string, and when we stimulate it, it shortens up because of the interaction that takes place inside on those microfilaments. All muscles that we're going to talk about share the same terminology, such as prefixes of myo and mice. Both refer to muscle, and sacro refers to flesh. So when we talk about sacromeres or myoglobin or those types of things, we're talking about muscles or flesh. So it's helpful to kind of know that terminology so you can help clear it up in your head when you get a little bit confused. Here's just a chart of the different types of skeletal muscles in our body, the different types of muscles in our body. I was looking at skeletal. Obviously, skeletal muscles attach to the skeleton of our skin. Cardiac muscles are specialized only to the heart, and smooth muscles are what we would find in our veins and our intestines and those types of things. Skeletal muscles we have control of. Cardiac and smooth muscles we don't. We can see below these, we can see individual pictures of what those muscles look like under a microscope. And you can see the lines in the first two. Skeletal and cardiac muscles are oftentimes referred to as striated muscles because we can see the striations in there, those lines in there. And those lines actually have to do with the way that muscles contract. And we'll talk about that more in depth and where those lines come from that creates that. Okay? So when we look at these cells, we can see that all of them have an endomysium. It's an outer lining around a group of cells. All of them share an endomysium, but that starts to get to where a lot of the similarities begin to end. But the endomysium actually is the part of the cell that attaches to whatever connecting point, maybe other cells, or with skeletal muscles, it's going to attach to the skeleton in the form of tendons and those types of things for attachment points. Okay, don't worry about that so much there. Most skeletal muscles are attached by tendons to bones. Most of them have multiple nuclei. By having multiple nuclei, they have a greater ability to repair and regenerate. They have the striation on there, or this visible banding, and we'll talk about where that visible banding comes from, but it comes from those myofibrils, or those muscle fibers inside of them that actually cause the muscle to contract. And skeletal muscles are voluntary, meaning they're subject to our control. Sometimes they seem to go a little haywire. If you get a muscle cramp in the middle of the night, I doubt you was wanting that, but they are under our control in general, and sometimes some of them share dual controls. These cells are surrounded by a bundle of connective tissue called the endomysium, and it encloses a single muscle fiber. So when we're talking about a muscle fiber, remember muscle fiber and muscle cell are the same thing. So we've got an endomysium that surrounds a muscle fiber, a paramysium that wraps around a fascicle or a bundle of muscle fibers. So we take a group of muscle cells, put them together, and we call that a fascicle. And around that, there's a paramysium. It's just another lining, a membrane. And then we have an epimysium. Remember, epi means above, that covers the entire skeletal muscle group, that whole mess of them, the whole muscle. And then, of course, the fascia is what's on the outside of the epimysium that kind of attaches it to other attachment points, such as the bone. So the epimysium goes around a single muscle fiber or a muscle cell. Remember, muscle fiber and muscle cell should be interchangeable. Paramysium is around a fascicle or a bundle of muscle fibers. Epimysium covers the entire muscle, and a fascia is what actually attaches it to other attachment items, such as a tendon or something like that. Okay? So try to stick with me on this. It'll hopefully make sense. If you get a little lost, a little confused, it's okay. Pause, back it up, play it back, or just let it play through. Hopefully when we get to some of these videos, it'll clear it up a little bit. But when we look at this picture, we can see that muscle fiber. It's just an individual cell. A group of those cells we call a fascicle, surrounded by the paramysium. And then we take a group of fascicles within and put them all together, and we wind up with a muscle. And that's surrounded by the epimysium. It surrounds the entire muscle. And then on down we've got the tendon that's attaching to the bone. Okay? The interesting thing about the epimysium that surrounds that entire muscle is ultimately it winds up blending into a connective tissue attachment of some sort. Usually it's a tendon. And tendons are really dense, really tough, strong cord-like structures that are made a lot of collagen fibers, which gives them a lot of strength and rigidity. They often cross a joint because of their toughness and their small size. It helps them be a whole lot more durable without taking up a whole lot of space. And that's why, like at the crook of your elbow, on the inside of your elbow, there's not a lot of muscle there. Well, that's because the tendons are coming in to attach there, and it saves on space, gives you more mobility, and you can close your arms farther than you would if it was muscle all the way to the bone. So it actually serves more than one purpose. It's an attachment point, but it also gives us a little bit of space too. And then this talks a little bit about the sheet-like structures that attach indirectly to the bones or cartilages. It's just another form of connective tissue. Okay? Muscles will attach to bones, cartilages, and other connective tissue coverings. So skeletal muscles will attach to a variety of things, but nothing should be too crazy, earth-shattering. When we move on to skeletal muscles, we're going to briefly talk about skeletal muscles and talk a little bit about cardiac muscles, but then we're going to come right back and talk about muscle cells and muscle structures and those kinds of things and how they work. Later on in consecutive chapters, we'll talk a little bit more about smooth muscles when we get into the digestive system, into the cardiovascular system. We'll obviously talk about cardiac muscle cells when we get into the cardiac section. So don't try to get too caught up in this other than just getting a general understanding of what a smooth muscle is and compared to skeletal or cardiac muscle. So skeletal muscles we just talked about, smooth muscles, smooth muscles don't have that striated appearance. They're kind of spindle-shaped cells, so they're odd-shaped cells. They're not long like a string. They're involuntary. We don't control them, which is a good thing because we'd have to sit around controlling our own blood pressure and our own digestive tract, which would probably take up much of our day and wouldn't be much fun. Found mainly in the walls of hollow visceral organs, like I said, the cardiovascular system, the blood vessels, and the digestive tract, your large and small intestines and your stomach and that kind of stuff. even a little bit in your urinary bladder and the respiratory passages. We can find it in there. And we see that the way that these muscles work, you can see most of the time we see these, they're going to be in this circular form like this. And they have this odd looking kind of elliptical, weird looking shape to them. But what that gives them is the ability, they kind of interattach to each other, and that gives them the ability to expand and contract and increase the diameter of this piece of the digestive tract that we're looking at here. So it can dilate and get larger to accommodate more food, or contract down and make it smaller to either push food through it or just to shrink back down in size when it's not full. So that's basically what they're going to do. Smooth muscles really are going to cause something to dilate or constrict. That's going to be the majority of what it is they do, and almost entirely really what they do for the purposes of what we're mainly talking about. Smooth muscles make your blood vessels dilate and your blood pressure get lower, or it's going to make your blood vessels constrict and make your blood pressure get higher, or it's going to push food through your digestive tract or help get urine from your bladder, or if you have a condition called asthma, it may cause the muscles that are surrounding the small air passageways in your lungs to constrict when you don't want them to, when they get in contact with some sort of allergen. It'll make them constrict as a reaction. And that's those smooth muscles that are doing that. Cardiac muscles, they do have striations, but they're similar in a lot of ways to skeletal muscles, but really they're very specialized and very different than skeletal muscles. We'll talk about that specifically in the cardiac section, but these guys usually have a single nucleus. They don't repair well. Remember, I told you that skeletal muscles have multiple nuclei and they repair very well. They're used to regenerating and repairing. Cardiac muscles are not that way. They're much more sensitive. They don't repair well by nature the way that they are. They are branching cells though, because the way they kind of branch out and interact, they're kind of an odd looking little cell, but they're really neat the way that they work. They join and communicate with each other through these intercalated discs, these contact points of where they make contact. And if you remember back a few chapters, we're talking about those intercalated discs where they look like they have wire fibers or hair fibers sticking into either side of the cell. That's a communication passageway to where that when we stimulate one cardiac cell, it causes all of them to react quickly, similar to a domino effect, only faster. Cardiac muscles are involuntary, thank goodness, and they are only found in the walls of the heart. The only place we're going to find them. So that's what makes them so specialized, is it's their location and their purpose. And what's neat is we can see the way that the muscles are wrapped around inside of our heart. They're specifically designed to push blood in specific directions. So when they do contract, they kind of squeeze in such a way that the atrium, the top part of the heart, will push blood into the bottom, and the bottom part of the heart will push the blood out into the body. So very specialized in the way that they work. Like I say, we'll talk about those other muscle groups more specifically in later chapters. Slide set B has a lot to do with the muscle groups of skeletal muscles on our body, and then we'll talk about the other groups, the other types of muscles. I'm sorry, I'm getting tongue-tied. We'll talk about the other types of muscles in their respective chapters when we get to them. We'll watch this little video about the 3 types of muscles. I know it's not really rocket science, but I thought this might be helpful to help keep us all on the same page.
Let's talk about muscles, and I've drawn the human body on the right, kind of a figure of it. And I want to talk about the 3 major types of muscles. And I thought it'd be helpful to have a picture, because then we can actually draw on there and show where the different types of muscles might be. So when I mention muscles, the word I want you to start thinking about in your head is movement. So think about all the different types of movements that might happen in your body. Just be really creative and start thinking of all the different movements. You might have, for example, a really easy one would be maybe, let's say your leg is moving. I'm going to just draw on our picture as we talk. But let's say your leg is moving because you're playing soccer. And so you've got this giant muscle in here, and this muscle is attached to a bone, right? There's a little bone here. I guess not so little, right? This is the largest bone in the body. It's called the femur. And so this muscle is attached to the femur, and this muscle is going to be attached by way of tendon. It's going to have tendons on both sides, and so this tendon is attaching it to the bone and allowing it to act on the bone. So this is an example of skeletal muscle, right? So this skeletal muscle is going to be attached to a tendon and bone. Now, that brings up the question, does every skeletal muscle have to be attached to a tendon and bone? Well, the answer is no, actually. There's some muscles that really aren't attached to tendons at all. In fact, right above the muscle we just drew is a muscle called the external oblique muscle. And don't worry so much about the names, but the idea here is that this muscle is actually not attached to a tendon. Well, in a sense, I guess you could think of it as a tendon, but it's like a flat tendon, basically a giant kind of sheet of fibrous tissue. And this fibrous tissue, is it kind of floating in midair? No, it's going to be connected to fibrous tissue on the other side because, of course, your body is symmetric. And so you've got fibrous tissue on the other side, and you guessed it, on the other side of that, you've got another external oblique. So you've got these muscles that are kind of coming into not really a tendon, but really a flat tendon, or something that looks like a flat tendon, and we call that an aponeurosis. So you might hear these words. I just want you to kind of be familiar with them. And now if someone asks you, is every muscle in the body attached to a tendon and bone? You can say no. Some are attached to a flat tendon called an aponeurosis. So the idea here is that you can kind of start identifying skeletal muscles, right? They're usually the muscles that you can see on your body, right? Actually, I don't even need to put quotes. That's the actual name for it, so no need for quotes there. So you can identify skeletal muscles pretty easily. But what about the other two? I mean, what about the cardiac and smooth muscle? I mean, you might wonder, is cardiac, does that mean heart? And is that the only type of cardiac muscle out there? And the answer is yes. This is your heart muscle right here. And the only type of cardiac muscle that we have in our body would be related to the heart. So in the heart, you can find specialized cells that were so interesting and different from skeletal and smooth muscles. They got their own name and category. So these are the cardiac cells, and you can only find them in the heart. So actually, I guess we're making a column of where you can find these cells. So what about smooth muscle? Where can you find smooth muscle? Well, for smooth muscle, think about any hollow organ, any organ that's got space on the inside, and blood vessels. Those are the two kind of major categories. Those aren't the only ones, but those are the major ones. That'll get you about 95% of the way there. So blood vessels and hollow organs are what you should think about. And hollow organs could be anything from, let's say, your stomach would be a hollow organ, or let me just put these examples here, or your bowels would be a hollow organ, anything like that. So I'm just going to write stomach here just to kind of jog your memory, where there's basically some empty cavity on the inside, right? And then as for blood vessels, just remember, I mean, one of the largest blood vessels, for example, is the aorta, and the aorta kind of comes up and over like that. And it's kind of like a hollow organ as well, right? I mean, there's a space on the inside of that blood vessel, and blood is usually flowing through that space, but at least it's hollow, so it's really not that different conceptually from the hollow organ. And just like in the hollow organ, the smooth muscle is in the walls of these things. So think about where the smooth muscle would be. It would be in the walls of the hollow organ or in the walls of the blood vessel. So that tells you where to find these different muscle types, right? And thinking about movement, smooth muscle can help the stomach, for example, move food forward. Cardiac muscle is going to help your heart beat. I mean, that's a pretty important movement. And skeletal muscle, I mean, we use that every single day. Every time you give your friend a high five or give your mom a hug, those are skeletal muscles that are helping your body move around, right? So let's move on. Let's think about some other differences between these categories. Let's talk about now the movement control. So who controls the movement, right? Do you control it, or is it automatically done? So smooth muscle is what I would consider automatic, or I'm going to call involuntary, because you'll probably see that word more often. Involuntary just means that your body is automatically taking care of it. And the same is true for your cardiac muscle. Involuntary, meaning you don't have to actually think about the next heartbeat. It just happens automatically, right? And skeletal muscle is the opposite. There it's voluntary, meaning if I didn't want to get up, then I would not get up, right? Or if I didn't want to go running, then I wouldn't go running. All of those movements of my body are under my control. I can decide when to do those things, right? And actually, maybe I'll draw little arrows here. What about speed? Which ones are fast and which ones are slow? So up here, the smooth muscle is the slowest, and the skeletal muscle would be the fastest. Which is pretty cool because the voluntary stuff, the stuff you control yourself, is the fastest. And the stuff that's happening automatically is pretty slow. And actually, it's nice because cardiac muscle is somewhere in between the two. It's somewhere in the middle. So when your blood vessels get tinier or they get big and vasodilate, all that stuff is happening on a pretty slow time scale as compared to, let's say, I jump and try to catch a ball. That's all happening really, really quickly. Thousands of little muscle movements are happening really lightning quick, and so those would be the fastest. Now, the final thing I'm going to draw is what these things look like. So how do they look? If you were to actually take a look at these cells, let's actually look at each of these one by one and figure out what they would look like. So the smooth muscle actually looks like a little eye. In fact, or like an almond, sometimes it's described that way. But I think of it as an eye, one single eye. And you can see that the edges or the ends are kind of tapered like that. And so sometimes you'll see that these are described as spindle-shaped. And I think that's kind of a holdover from a time period long ago when people thought about spindles more than they do now. And the other thing, it's got one nuclei, right? It drew that right in the middle, one nuclei, and it's in the middle of the cell. So that's basically what a smooth muscle cell looks like. What about a cardiac cell? Well, this cell is branched. That's actually one of the most interesting hallmark features of it. Now, not every single cardiac cell is branched. Some are actually just kind of humdrum, looking normal, maybe like this. But the fact that you can find branched ones is what really makes these so easy to recognize, right? And if you look at a whole bunch, I'm going to erase this guy now that you know he exists, but I'm going to focus on the branched one because these are the ones that make them very easy to spot. And they also have nuclei, sometimes one, but sometimes two, which is interesting because, usually you think, oh, well, one cell, one nuclei. But the reason I had to point that out for the smooth muscle cell that there's only one is that sometimes these cardiac cells have more than one. So the two features, I'm going to just write out here, branched and one or two nuclei. So not always two, but they can have two. And they're also located kind of in the middle of the cell. And I'll show you what I mean by middle when I draw the skeletal muscle. I'll do that now. This is the skeletal muscle, and it's got something like this. It's got these little outpouchings I'm trying to draw for you, and you'll see in just a second what I'm drawing. These are little spots on the edge or on the periphery for nuclei. And notice that there's not one nuclei, not two nuclei, but bunches of nuclei. So these cells are actually working as a giant cell in a sense. So these are actually, first of all, they're straight. They're not branched. So straight. And they've got many nuclei. This is actually really, really important. And you can see how it would be easy to spot these guys, right? Because they've got many nuclei, and the nuclei themselves are in the periphery, kind of on the edges. And that's why I wanted to point out that the other two are in the middle. Now, kind of a final point is that if you were to look at these under a microscope, and actually this is something that was noticed a long time ago, they would look something like this. And this is called striated. So they basically have these striations, but notice that the smooth muscle cells don't have this. It's really just the skeletal muscle and the cardiac muscle that has these striations. And so sometimes you'll hear about striated muscle, and they could be talking about either of the two, right? They could be talking about cardiac or skeletal, but you know that they're not talking about the smooth muscle. So this is striated. And striated just kind of refers to those stripes. And that's what it looks like under a microscope. And we'll talk about exactly why they're striated and what that would imply about the cell in another video. But I just want you to get a kind of a rough lay of the land. And now you can see there's actually some interesting stuff here. You have some similarities between the cardiac and the smooth muscle. They're both involuntary. You've got some similarities between the skeletal and the cardiac. They're both striated. And so you can see how all three are somehow similar, but also somehow different from one another.
Okay, so now we should all be on the same page about the different types of muscles that are in our body and have a better understanding of how the three of them work. Now, remember, we're going to talk about the other two a whole lot more in depth, cardiac and smooth, when we get to those chapters. But at least we've kind of got a good starting point, okay? So, whoops, there. It's wanting to go 2 slides at a time. You guys might like that in the short term, but it wouldn't be good in the long run. All right, so the functions of skeletal muscles, of course, they provide movement, which is kind of obvious. Maintain posture, sometimes you don't really think about, but setting up in a chair or standing up, it requires different muscles at different times to be either tense or relaxed in order for us to maintain our balance. That's why it's so difficult for newborns to learn how to walk, because there's so much that's involved with them learning how to walk or stand or sit up or do those types of things. There's so many different muscles, involved in that happening. They also help stabilize joints, keep our joints from coming apart, which would not be good. Sometimes they do anyway, playing football or basketball or doing some things that we shouldn't be doing or that's rough on us that we're too old to do. They also generate heat. They're a great source of heat that helps keep our blood warm and our bodies warm, which is a good thing. Most of the time, they can't actually generate too much heat, and that can be problematic. Now, if we take a closer look microscopically at a skeletal muscle, and remember what we're primarily going to be talking about here in this next section when we're talking about how these muscles work is skeletal muscles. So we look at them microscopically. We see there's something called a sarcomina. I never can say it right. Sarcomia. You get it, right? It's just a specialized plasma membrane on the outside of each skeletal muscle cell. So this membrane covers the outside of it, a specialized membrane for skeletal muscles. I don't know why they decided to begin changing the names of these membranes. Once we finally figured out what cell membranes were and all that, they decided to throw new names in there. But basically serving a very similar purpose of, the plasma membrane that we were used to calling it, but since it's a sarco, it's a tissue membrane, so sarcolemma is a tissue membrane. We also have things that are called myofibrils, or muscle, myo meaning muscle, myofibrils. They're long organelles. Remember the little organs inside of a cell, we call them organelles. This is a new organelle. We haven't talked about this one yet, but they're called myofibrils. They're long organelles. They're inside of muscle cells. And they have these different bands, these light I bands and these dark A bands that give it that striped appearance, that striated look that we're seeing in there, is these bands that we see. And we'll talk about those more in depth here in a little bit. But so when we take a look at this, now we're looking at one muscle fiber or one muscle cell. And if we look inside of there, we see all of these myofibrils. These are little organelles. They're like, remember we talked about mitochondria and nuclei and all of those things in the other chapter. These are organelles that are specialized to muscle cells, skeletal muscles. They have all these myofibrils, and you see these lines and striations on there. Those are the dark and light bands, and if you, over here on the left, it's showing them the dark and light bands. Those are dark and light bands are going to become important later because we're going to talk about how they interact to make a muscle contract. That myofibril is the working unit of skeletal muscles. It's where the contraction happens. It's how muscle cells are able to shorten. Remember this one picture that we're looking at, this big tube here with all these myofibrils in it, that is one muscle cell, okay? So we're really zoomed in on it. And then we look at this banding, this pattern banding We have this I band, which is a light band, and it's spelled with an I, so that helps us remember that a little bit. And it only contains thin filaments. And then, of course, there's a midline interruption called a Z disc, and then we have the A band, which is the dark band, and it contains the entire length of thick filaments. So I band is thin filaments, A band is thick filaments, and then on the A band there's an H and an M zone. We'll talk about those zones a little bit more, and we'll talk about those bands, but I don't want you to get caught up in trying to memorize what all of those are. You're going to need to be familiar with them, at least passively familiar with them. But I don't really like testing on these just because I don't want your focus to be on the bands, battle of the bands. I don't want you guys to be focused on that stuff when there's other stuff in here that I think it's much more important that you come away with. It's very easy to think that this is the important stuff, and I don't want you to think it's not important. It is important in the context of the bigger picture of what we're going to learn, that you understand that these bands exist, but being able to pick them out of a lineup, I don't think is something that you need to be very critical about, okay? So just bear that in mind. We take a closer look at, remember, this is the inside part, this is one of the organelles inside of that skeletal muscle, and it's the myofibril, sometimes called the fibril. And within that, we have a space within that called a sarcomere, okay? So lots of new words. But that sarcomere is where those bands overlap, okay? So we've got an A band in the middle, and you can see the A band has thicker filaments there in red. We can see the red color of those, where those are thicker filaments, right? We can see right here, these are the A bands. Okay, and the I bands here, this is where the thinner filaments are, okay? And then you see how they overlap with each other. See, we've got this overlapping of the A bands and the I bands. And this one section right here, this one section of where these overlap is called a sarcomere. Now, right down here, it's cut off, but there's another sarcomere over here, and there's another sarcomere over here. So there's multiple sarcomeres down the length of a myofibril, okay? And what happens at these points, where these overlap right here, these manage to interconnect in a way that actually pulls them together. So these will interact in such a way to where that these actually begin to pull together. Where they're setting like this, they'll pull together. See if I can put myself up on the big screen. Where they're setting kind of like this or overlapping, when we stimulate that cell, they'll pull like this together. They'll pull overlappingly as far as they can to where that that muscle cell will contract. It will shrink. It will get shorter, and that's where that happens is in the sarcomeres, in those in the A and I bands. And then, of course, you can look and you can see the Z disc and the H zone and the M line, and you can see all those in there where there's just identification features of what those are called. Like I said, don't get caught up in all of that. It's very important that you understand that the sarcomere and that's really the functional unit of a myofibril. That's where all the action is taking place. Okay, so bear with me. If you feel like you're getting lost, just bear with me, and let's keep moving forward and get through it. And I think by the time we get to the end, at least I hope so, you'll have a pretty good understanding. Okay, so the sarcomere, like I said, it's that contractile unit of the muscle fiber. So it's that one section that contracts, but remember there's multiple sections down the length of that are going to be contracting. And how this is organized is, of course, by thick and thin filaments. Now, you got thick filaments. Remember, thick filaments is the A line, right? Thin filaments is the I line. Thick filaments should be associated with myosin filaments, okay? So when we think thick, we want to think myosin and thick. Those two go together. Thin and actin, those two go together. So I'm pointing those out, and I'm making the link between them two is eventually I'm not going to be calling them thick and thin. I'm going to be calling them actin and myosin, okay? So when I'm talking about actin and myosin, I'm still talking about the thick and thin filaments, okay? And I want you guys to realize that So when I change my terminology, you guys are still with me. So actin and myosin are those thick and thin filaments that we just looked at on the previous slide. These bands that we're talking about are actin and myosin bands. That's what those are. And they're probably not ones you're going to go see at Oktoberfest or anything anytime soon. Of course, it would be interesting to call your band that. The I bands. No, sorry. It's as good as it gets. I'm not very good at jokes. So, the thick filaments contain myosin fibers or myosin filaments, and they're made up of a protein, oddly enough, called myosin. Go figure. They contain ATPase enzymes. They possess myosin heads, which we'll look at them here in a minute, and you'll see what myosin heads look like. And these heads are also known as cross bridges because they are the link between the thick and thin filaments. Remember how I told you that those two filaments are going to interact and they're going to pull together to get shorter? It's the myosin filaments, the head of the myosin, that actually cause that pulling together or that contraction to take place. Thin filaments are the actin filaments. They contain a contractile protein called actin, and it's anchored at the Z disc. If you want to look at that picture and go, hey, where's the Z disc? And that's where those things are anchored to. At rest, they lack actin filaments, and that's called the H zone there, where there's that lacks those filaments when they're at rest. But don't get too caught up in that. All right. This is a pretty good picture right here. This is going to help clear things up. Remember that sarcomere that we talked about that was inside of that myofibril? That one section of it, remember this one section? Remember there's multiple sections, hundreds or thousands or however many sections down the length of a muscle cell within that myofibril? Well, you can see the little heads on here, right? You can see those little myosin heads on top of the myofilament, or on the thick filament, and then the thin ones is the actin. So these are actin, and these are myosin. And these doggone things interact with each other like nobody's business. Okay? The way that they work, these little actin heads, these little myosin heads, they will make contact on the actin side. And then once they make contact with an actin site, they will automatically, like somebody lifting barbells, when they make contact, they'll automatically, like somebody lifting barbells, pull like this. So you've got that filament that's over the top here, and it's going to pull that filament back. So it's going to pull it tighter, and that's what those little actin heads do. What's interesting is once we stimulate them, they don't just pull one time. Oftentimes it's like a ratchet. There's all of them things are in there doing this. They're pulling. They're constantly pulling it until the muscle can pull no tighter, and then they're just holding it there. If we're holding that muscle real tight and rigid, they just keep ratcheting and holding it in place and holding it there. They don't just pull and stay. It's a constant ratcheting motion to hold it in, but it's going to take a constant nerve impulse in order for that ratcheting action to occur. So that's what's happening in that site. Whenever we stimulate the muscle to contract, those actin and myosin filaments come in contact with each other and they pull together. They will pull this whole thing. They will pull those right there. They'll pull together, and it's hard for me to do it. I don't know if I can do it where you can see it. They'll pull together like this. Okay? So that's how they'll work. I've got a couple of videos that we'll look at here in a little bit that will help clear that up and explain it a little bit better, and hopefully the repetition of that will help. So let's look at the anatomy of this first, and then we'll talk about how the function of it works. So we're going to look at the anatomy and explain it a little more. I do apologize that the Khan Academy videos are not as exciting and over the top as some of the others, but they're very, very informational and they're very, very accurate. So let's watch this video here and hopefully get us all up to speed.
I think we have a respectable sense of how muscles contract on the molecular level. Let's take a step back now and just understand how muscles look, at least structurally, or how they relate to things that we normally associate with muscles. So let me say I draw a flexing bicep right here. So that's someone flexing their bicep. That's their elbow, and let's say that's their hand right there. So this is their bicep, and it's flexing. So this is their bicep. I think we've all seen diagrams of what muscles look, at least on kind of a macro level. And it's connected to bones at either end. So let me draw the bones. I'm not going to detail where. So it's connected to the bones at either end by tendons. So this right here. So right here would be some bone. Right there would be another bone that it's connected to. And then this is tendons, which connects the bones to the muscles. So this right here is tendon. Tendon. And we have the general sense connected to two bones. When it contracts, it moves some part of our skeletal system. So we're actually focused on skeletal muscles. Skeletal. The other types are smooth muscles and cardiac muscles. Cardiac muscles are those, as you can imagine, in our heart. And smooth muscles these are more involuntary, slow-moving muscles in things like our digestive tract. And I'll do a video on that in the future. But most of the time when people say muscles, we associate them with skeletal muscles that move our skeletal system around, allow us to run and lift and talk and do and bite things. So this is what we normally associate. Let's dig in a little bit deeper here. So if I were to take a cross section of this bicep right there, if I were to take a cross section of that muscle right there, so let me do it big. So if this is the cross section, so the bicep or that muscle, I'll stop saying biceps because I want to be general. So I'm going to take a cross section of it. So this is the cross section. This is where I've taken my cut. And then it'll look something like this. This is the inside of this muscle over here. Now, I said back here we had our tendon. Back here we had our tendon. And then there's actually a covering, and there's no strict demarcation or dividing line between the tendon and the covering around this muscle, but that covering is called the epimysium. Epimysium, and it's really just connective tissue that covers the muscle, kind of protects it. It reduces friction between the muscle and the surrounding bone and other tissue that might be in this person's arm right there. And then within this muscle, you have connective tissue on the inside. Let me do it in another color. I'll do it in orange. This orange tissue right here. And let me make this orange tissue, and then it divides off little, I guess we could call them fibers of some sort. So we have this orange tissue right there. This is called the perimysium. And that's also just connective tissue inside of the actual muscle. Perimysium. And then each of these things that the perimysium is dividing off. So each of these things at the perimysium, let's say if we were to take one of these things and allow it to go a little bit further. So if we were to take this thing right here, what this perimysium is dividing off, and if we were to pull it out, actually, let me do this one right here. If we were to pull this one out, just like that, so you have the perimysium surrounding it, right? This is all perimysium, and it's just a fancy word for connective tissue. If perimysium, and there's other stuff in there. You could have nerves, and you could have capillaries, all sorts of stuff, because you have to get blood and neuronal signals to your muscles eventually. So it's not just connective tissue. It's other things that have to be able to eventually get to your muscle cells. So each of these, I guess you call it subfibers, but these are pretty big subfibers of the muscle. This is called a fascicle. This is a fascicle right here. So that right there is a fascicle. That's a fascicle. And then the connective tissue inside of the fascicle is called the endomysium. Endomysium. So let me draw the endomysium right here. So once again, more connective tissues, has capillaries in it, has nerves in it, all of the things that have to eventually come in contact with muscles, with muscle cells. We're inside of a single muscle. So let me draw the endomysium. So all this green connective tissue is endomysium. Endomysium. And each of these things that are in the endomysium are an actual muscle cell. This is an actual muscle cell. So I'll do it in purple. So this thing right here, I can pull it out a little bit. Let me pull this thing out a little bit, just like that. So if I pull this out, this is an actual muscle cell. This is what we wanted to get to, but we're going to go even within the muscle cell to understand how all the myosin and the actin filaments fit into that muscle cell. So this right here is a muscle cell, or a myofiber. Myofiber. The two prefixes you'll see a lot when dealing with muscles. You're going to see myo, which you could imagine refers to muscle. And you're also going to see the word sarco, like sarcolemma or sarcoplasmic reticulum. So you're also going to see the prefix sarco, and that's flesh. So, sarcophagus, or you could think of other things that start with sarco. So sarco is flesh, but when muscle is flesh and myo is muscle. So this is myofiber. This is an actual muscle cell. And so let's zoom in on that actual muscle cell. So let me actually draw it really a lot bigger here. So an actual muscle cell, it's called a myofiber. It's called a fiber because it's longer than it is wide. And they come in various, so let me draw the myofiber like this. So this is my muscle cell right here. And I'll take a cross section of the muscle cell as well. So this is my myofiber. Myofiber. And these could be, relatively short, several hundred micrometers, or it could be quite long, or at least quite long by cellular standards. We're talking several centimeters. So several centimeters, if you think of it as a cell, that's quite a long cell. And because it's so long, it actually has to have multiple nucleuses. And actually, to draw the nucleuses, let me do a better job drawing the myofiber. I'm going to make little lumps in the outside membrane where the nucleuses can fit on this myofiber. Remember, this is just one of these individual muscle cells. And they're really long, so they have multiple nucleuses. And let me take its cross section because we're going to go inside of this muscle cell. So I said that it's multinucleated. So if we kind of imagined its membrane being transparent, then there'd be one nucleus over here, another nucleus over here, another nucleus over here, another nucleus over there. And the reason why it's multinucleated is so that over large distances, you don't have to wait for proteins to get all the way from this nucleus all the way over to this part of the muscle cell. You can actually have the DNA information close to where it needs to be. So it's multinucleated. I read one, I think it was 30 or so nucleuses per millimeter of muscle tissue is what the average is. I don't know if that's actually the case. But the nucleuses are kind of right under the membrane of the muscle cell. And you remember what that's called from the last video. The membrane of the muscle cell is the sarcolemma. 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 Or sarcolemma, however you want to call it. These are the nucleuses. The nucleuses. And then if you go even within, if you take the cross section of that, there are even tubes within that. There are tubes within that called myofibrils. So here, I could take, there's a bunch of tubes inside of the actual cell. Let me pull one of them out. So I've pulled out one of these tubes. This is a myofibril. Myofibril. And if you were to look at this under a light microscope, you'll see it has little striations on it. You'll see it has little striations. So the striations would look something like that, like that, like that. And then there'll be little thin ones like that, like that. And this is inside of these myofibrils is where we'll find our myosin and actin filaments. So let's zoom in over here on this myofibril. We'll just keep zooming until we get to the molecular level. So this myofibril, which is, remember, it's inside of the muscle cell, inside of the myofiber. The myofiber is a muscle cell. Myofibril is you can view it as a tube inside of the muscle cell. And these are the things that are actually doing the contraction. So if I were to zoom in on a myofibril, you're going to see it. It's going to look something like that. And it's going to have those bands in it. So the bands are going to look something like this. You're going to have these little short bands like that, like that. Then you're going to have wider bands like that, like these little dark thing. Let me trying my best to draw them relatively neatly. And there could be a little line right there. Then the same thing repeats over here. So each of these units of repetition, is called a sarcomere. This is a sarcomere. Sarcomere. And these units of repetition go from one, this is called a Z line, to another Z line. And all of this terminology comes out of when people just looked at it on a microscope and they saw these lines. They started attaching names to it. And just so you have the other terminology, we'll talk about how this relates to the myosin and the actin in a second. This right here is the A band. And then this distance right here, or these parts right here, these are called the I bands. And we'll talk about really in a few seconds how that relates to the mechanisms or the units that we talked, or the molecules that we talked about in the last video. So if you were to zoom in here, if you were to go into these myofibrils, if you were to take a cross section of these myofibrils, what you'll find is, if you were to cut it up, maybe slice it this well, it's hard to, if you were to slice it parallel to the actual screen that you're looking at, you're going to see something like this. So this is going to be your Z band, which is really just, so that's your Z band. This is your next Z band. So I'm zooming in on one sarcomere now. This is another Z band. Then you have your actin filaments. Now we're getting to that molecular level that I talked about. So you'll have your actin filaments. I'll draw them like that. I'll just draw a couple. You have your actin filaments that look like that. Let me label it. So these are the actin filaments. These are the actin filaments. And then in between the actin filaments, you have your myosin. Filaments. So let me draw it. Myosin. So let me draw it in this color. Remember, the myosin filaments had those two heads on them, right? They each have two heads, and they two heads like that crawl along the actin filaments. I'm just drawing a couple of them, and then they're attached at the middle, just like that. And we'll talk about in a second what happens when the muscle actually contracts. And I could draw it again over here. So it has many more heads than what I'm drawing, but this just gives you an idea of what's happening. So these are the myosin, I guess, proteins, and they're all intertwined like we saw in the previous video. And then there'll be another one over here. I don't have to draw it in detail. So you can see immediately that the A band corresponds to where we have our myosin. So this is our A band right here. A band. And there is an overlap, right? They do overlap each other even in the resting state. But the I band is where you only have actin filaments, no myosin. So this is the I band right there. And then the myosin filaments are held in place by titin, which is you can kind of imagine as a springy protein, by titin. Now I want to do it a different color than that. Let me do it in, so that right there. So that's the myosin is held in place by titin. So that right there is titin. It's attached to the Z band by titin. So what happens? So we have all of these. When a neuron excites, so let me draw an endpoint of a neuron right here, the endpoint of an axon of a neuron right there. It's a motor neuron. It's telling this guy to contract. You have the action potential. The action potential travels along the membrane, really in all directions. And then eventually, if we look at it from this view, there you have those little transverse or T tubules that essentially go into the cell, and continue to propagate the action potential. Those trigger the sarcoplasmic reticulum to release calcium. The calcium attaches to the troponin, that's attached to these actin filaments. That moves the tropomyosin out of the way. And then the crawling can occur. The myosin can start using ATP to crawl along these actin filaments. And so as you can imagine, as they crawl along, let me switch colors. Their power stroke is going to push. They're going to push you could either view it as the actin filaments in that way, or you can say that the myosin is going to want to move in that direction. But you're pulling on both sides of a rope, right? So the myosin is going to stay in one place, And the actin filaments are going to be pulled together. The actin filaments are going to be pulled together, and that's essentially how the muscle is contracting. So we've hopefully in this video connected the big picture from the flexing muscle all the way over here to exactly what's happening at the molecular level that we learned in the last few videos. And you can imagine when this happens to all of the myofibrils inside of the muscle, right? Because the sarcoplasmic reticulum is releasing calcium generally into the cytoplasm of, which is also called myoplasm because we're dealing with muscle cells, the cytoplasm of this muscle cell. The calcium floods all of these myofibrils. It's able to attach to all of the troponin, or at least a lot of the troponin, on top of these actin filaments, and then the whole muscle contracts. And then when that's done, each muscle fiber or myofiber or each muscle cell will not have that much contracting power, but when you couple it with all of them that are around it, if you just have one actually working or a few of them, you'll just have a twitch. But if you have all of them contracting together, then that's actually going to create the force to actually do some work or actually pull your bones together or lift some weights. So hopefully you found that mildly useful.
Little better idea. And I know he kind of at the end started talking about some other things that we haven't discussed yet, as far as the specific details of some actin and myosin and how they interact and the role that calcium plays in that. We're getting to that. He talked about troponin and tryptomyosin and their role in that. We're going to get to that too. My main thing that I wanted you to catch was that first part, but I didn't want to cut him off in case you were kind of getting that. So we're going to talk about the sarcoplasmic reticulum. You remember the endoplasmic reticulum that we talked about in the other cells earlier on? There's one of them inside of muscle tissue. We're going to name it a new name because it's muscle tissue and not another type of cell. We call it a sarcoplasmic reticulum. And it doesn't have, it doesn't create proteins, so it doesn't have ribosomes on it, so it's all smooth. But it's a different form of smooth reticulum. So that sarcoplasmic reticulum that I've talked about, it's a specialized endoplasmic reticulum, a little different than what we talked about earlier, because its main job is to store and release calcium at certain times when it's told to. So when the nerve impulse comes down, it causes the release that he talked about, that electrical impulse coming down from the nerve, stimulating the membrane of that muscle cell, and causing that to transmit across the membrane and down those transverse tubules. We're going to talk about them in a minute. But that's how that nerve impulse gets to the endoplasmic reticulum, that sarcoplasmic reticulum, to release the calcium. And because that sarcoplasmic reticulum surrounds all those myofibrils, all those organelles, you know, those fibers that run the length of it that have the sarcomeres on them, that reticulum, that sarcoplasmic reticulum surrounds that, and it's full of calcium. And we'll talk about how that works. So first thing, let's back up and let's talk about how we stimulate a contraction, a muscle cell to contract. So one of the terms that we need to be familiar with is irritability. It's an ability for something to receive and respond to a particular stimulus. Muscle cells have an irritability factor, meaning they have the ability to respond to an impulse. When they receive a stimulus, they can respond to it. They also have some contractibility. They can shorten. Remember, we've just talked about that. You know, along the thick and thin filaments with actin and myosin, that's how they're going to contract and shorten. And they also have some ability to be kind of stretched a little bit too. We see them resting at a certain point, but we can stretch them a little bit and lengthen that out, that distance between the thick and thin filaments. We can stretch that out just a little bit, but they're elastic and they'll recoil back to their normal resting state once we're done. Those are some unique features about skeletal muscles. But in order for them to work, the doggone things have got to receive a signal from somewhere. So it just doesn't magically communicate. Unfortunately, even though your cell phone in your pocket is wireless, your body is not. Your body has to be wired. There needs to be a nerve connecting to every single muscle in order for that muscle to function. So that's how muscles are stimulated to contract. If we put a group of muscles together and have one nerve cell going to it, we call that a motor unit. And that's typically how that works. We don't have one nerve coming from the brain to stimulate one muscle cell. Otherwise, we would be just a bundle of nerves and very few muscles. So to combat that, the way that it works is we have a group or a bundle of muscle cells together, or a unit of muscle cells together that when we stimulate that group with one nerve, that whole group of muscle cells are going to contract. So that way we don't have to have a nerve going to every single individual muscle cell in the body. It can go to groups of muscle cells and be able to do the tasks that we want. And we can see that here where we've got two different nerves actually going to this group of muscles, but they're going to serve two different purposes. But basically, and that's kind of a simplistic way of looking at it. It's probably a whole lot more muscle cells involved in this this motor group than what that's depicting. But we can see an example of it there, where the nerves are actually making contact with those muscle cells. So now we'll have one nerve that comes down, and it's going to branch out and make contact with all of those muscle cells, individually. Okay, so it's not hey, I'll stimulate this muscle and the one next to it will contract too. No, each individual muscle cell needs to be stimulated by that same neuron. Cardiac cells are different, whereas if we stimulate one cell, it will go like dominoes to the rest of them. Skeletal muscle cells are not that way. They each have to be innervated, but one nerve can stimulate hundreds of muscle cells. It can branch out and make contact with hundreds of muscle cells. And that picture right there shows that branching down here at the bottom, where that's just branching out in like a tree, just branching out from one nerve. Where that nerve makes contact with that muscle, we call that the neuromuscular junction. And it's important that we look at this neuromuscular junction and we study it a little bit more, because it's going to help us to understand these a little bit more. So just because that nerve makes contact with that with that muscle doesn't necessarily mean that that's the end of it. That's just a simple connection, like plugging your blow dryer into an outlet and all of a sudden everything works. There's not a physical connection between the nerve and the muscle cell. There's a gap in between there. So when we look at this neuromuscular junction, there's actually a gap. So, and I'm trying to find me a clear space I can write on. But we've got our muscle cell down here, and we've got this nerve that comes down and that gets right up next to it, and there's actually a gap in between it. And that's going to erase the whole thing probably, yeah. And there's this gap kind of that comes up, and it's a little dip there maybe, and then goes around it. So we've got a gap in between the nerve and the muscle cell. So how are we going to get the impulse from the nerve to the muscle cell in order for that muscle to contract? Well, we do that by chemicals. And these chemicals, we refer to them as neurotransmitters, because it's bringing a nerve impulse in and transmitting it to a target organ. And in this case, it's a muscle cell. So we have to have a chemical that's released by the end of the nerve, by the terminal end of the nerve, that can be sensed by the muscle cell on the other side, or the organ on the other side, so that it will react. With the skeletal muscle system, with skeletal muscles, that neurotransmitter is acetylcholine. That's an important thing to remember. Write that down. Acetylcholine is the neurotransmitter for skeletal muscles. And I'll explain that in just a little bit more here in a second of how that works, okay? Because right there, we've got that, what we call a synaptic cleft, or the gap between the nerve ending and the skeletal muscle. We've got that gap right there. And there's no contact there, but there's some fluid in between there. There's some interstitial fluid. It's a watery matrix right there. When that nerve impulse comes down and it reaches the end of that nerve, the very end plate, or the axon terminal of that motor neuron, there's some channels in the end of that are going to open up. The stimulation of that membrane on the outside of that nerve is going to cause these calcium channels to open, and calcium is going to enter the end plate of that neuron, of that nerve cell. That calcium coming in is going to trigger the presence of calcium in that neuron is going to trigger the release of acetylcholine, that neurotransmitter I just talked about. That acetylcholine is going to go across that little gap between the nerve and the muscle and make contact with the membrane on the other side, makes contact with that muscle membrane, and that muscle membrane is going to react to the presence of acetylcholine. Now, I realize I just went through a whole lot of things, and it sounds like a whole lot, but let me see if, well, I'm going to see if there was, I was going to see if there's enough space where I could kind of draw that out, but I'll draw it out for you, or at least we'll have a picture of it here in a minute. Once there's enough acetylcholine released across there, it's going to cause that membrane to react. The membrane's going to react. Sodium's going to rush into the cell, and potassium's going to leave it. And the membrane's going to respond to that, and that impulse of that's going to spread throughout the entire length of that muscle membrane. It's even going to go deep down inside of that membrane, and it's going to cause the channels inside of there that's holding that calcium, that sarcoplasmic reticulum, to open to release its calcium, because it's causing the whole thing to depolarize because it's allowing that in. Now, in order for that contraction to not continue on endlessly, there needs to be something that's going along and taking up the acetylcholine. As long as there's acetylcholine making contact with the membrane of that muscle cell, it's going to keep trying to contract. So there's a substance that's present right there in that junction, in that space in between. There's this acetylcholinesterase, and its job is to break down acetylcholine. Whenever acetylcholine's there, it's going to try to gobble it up. So acetylcholine is released by the nerve, and immediately it's starting to be recaptured by acetylcholinesterase, but some of it makes contact with the other side, and it causes the nerve or the muscle membrane to contract, or it stimulates it. And when that happens, that acetylcholinesterase begins taking it back up pretty quickly too, so the impulse is short-lived. It happens quickly. But if there's a continual nerve impulse of that coming down, acetylcholine is going to continually to be released. Acetylcholinesterase will keep trying to take it up, but it can't keep up as long as there's more being released. Once acetylcholine is no longer being released from the nerve, acetylcholinesterase will take all that up, and the stimulation of the muscle membrane will go away, and the muscle will stop contracting. Okay, so that's kind of how that works. And then the muscle cell will begin to return back to its resting state by pumping sodium, potassium back out of it to try to make that membrane back at what we call a resting potential. So the membrane, we talked about this a few chapters back, the sodium potassium pump, and how it sets up the membrane on the outside of a cell to be reactive to a stimulus. And the way that it reacts to a stimulus is it opens up channels, and sodium and potassium rush in and out of it. And then it's got to reset by pumping it back down to the level that it wants, because it's pumping it against the normal diffusion gradient. But that's what it's doing. It's trying to set that up to where that cell membrane is sensitive to an impulse. So it sets it up to where it's sensitive to an impulse, and this particular impulse happens to be acetylcholine that sets it off. That's the neurotransmitter that it likes. So we can look at this picture, and finally we've got a picture to kind of look at, and hopefully I can help make a little bit of sense of it. But we've got this picture, and we see this is the nerve right here that we're talking about, and this action potential, and I know we haven't talked about action potentials yet. We'll talk about this a little bit more later when we talk about nerves, but basically it's the impulse that's coming down, the nerve signal that's being sent down to the end of this nerve. And what happens when this action potential reaches here, and the action potential has to do with the stimulation of this membrane on the outside of the nerve, and that's how the action potential actually moves down the nerve. But once it reaches down here at this point, this stimulation of this causes these calcium channels to open up. They open up and allow calcium to rush in to the end of this neuron. And when the presence of calcium in the end of this neuron, it causes this neuron to release this acetylcholine. All right, you can see the acetylcholine being released down here. Calcium comes in, it triggers the release of acetylcholine. Acetylcholine comes out and it comes across, and it attaches to these channels over here, and it causes these channels to open up, and that goes across the whole membrane. Eventually, it gets down here in the sarcomeres and causes them to contract. So that's how the whole process works. Now, acetylcholinesterase is going to come along, and it's going to try to reuptake all of this acetylcholine as quickly as it's released. So you can see how some make contact and some are still floating around in there. That's because acetylcholinesterase is kind of trying to take them up. And acetylcholinesterase will actually take them up and bring them back up here and give them back to the end of the neuron. So that's how that nerve impulse happens and how we stimulate the membrane of a muscle cell. So that's all we've done. We've had a nerve impulse come down, stimulate the membrane of a muscle cell by releasing acetylcholine. Remember, it's the acetylcholine that actually stimulates the muscle cell to open up its calcium or sodium and potassium channels. So it opens up those channels and it causes that whole membrane to depolarize. And when it depolarizes, it sends that signal across the whole membrane. And in the process of doing that, it goes down deep inside of that muscle cell, that muscle cell. It goes down deep inside of it and triggers things to happen at this level, down here at the sarcomere level. Okay, nerve impulse comes in. It's stimulated the membrane on the outside of the muscle cell, and now it's going to make things happen on the inside. So, and that's just going through that exact same thing that I just drew. It's going through it step by step. Come on. Doesn't want to change slides now. There it goes. Okay. So the way that it describes the stimulation of that membrane is kind of taking a match, lighting a twig. It starts in one spot, but it rapidly goes down the whole length of it. So when we stimulate that here with this nerve, it's going to go all down the length of it. And we're just talking about the membrane on the top. So it's going down the membrane on the top, and as it's going along, it's actually going inside too when it's going along on down this membrane. But it's traveling the length of the membrane and then getting inside too, causing a reaction to happen inside of that muscle cell. Okay? This video is pretty good, and it's not real long. So let's just take a peek at it.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in two ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Clears that up just a little bit better than what I was trying to do. It's difficult to do without having you in the room with me, because it's easier for me to be able to answer some questions and clear things up that you may not be getting. That's why I'm trying to throw the videos in here to be as helpful as possible. So, like I say, don't hesitate to continue to go out and look for resources. I'm betting right now you're probably struggling with action potential, with what they're talking about with action potential and the gated channels and the influxes causing a change in the membrane because of the voltage changes. Feel free to go out and look that up. We will talk about it more later when we talk about neurons, but just really just kind of get it in your head that stimulation of that nerve, or I'm sorry, the stimulation of that membrane that's covering the cell, the muscle cell, causes an excitement in that membrane that makes something happen, and we call that an action potential. We're going to talk more about it later, so try not to get too caught up in that, okay? What happens then is that action potential, that excitement along the membrane, makes its way inside of that cell, and when inside of that cell, it releases calcium. And I know the slides aren't talking about this, but it causes that excitement along the membrane. There's some tubules, there's some holes, some little tubes along the way down that muscle cell that allow that excitement of the membrane to travel inside of the cell. And it triggers, that excitement goes down there and it triggers the release of the calcium that's stored in the sarcoplasmic reticulum. That sarcoplasmic reticulum is, it's like an endoplasmic reticulum, but it surrounds all the myofibrils, all those fibrous organelles inside of there that do the contracting. Well, it's that cistern that it's called of calcium that's stored in those is released into those myofibrils. What happens is when that calcium is released into the myofibrils, the calcium is, troponin is heavily attracted to calcium. So, and the troponin is holding the tryptomyosin on the myosin sites so that actin can't touch it. And I'm sorry if it's very confusing, but it'll become clear here in a minute. So actin and myosin are actually protected from touching each other. They've got like a chaperone surrounding them. And it's myosin that has the chaperone surrounding it. It's kind of twisted around it, kind of lazily twisted around it. And it's called tryptomyosin. Tryptomyosin is actually stuck on the myosin site so that the actin head can't touch it. It's like trying to keep two teenagers apart, right? So we've got a chaperone in there. So tryptomyosin is the chaperone that's protecting the two of them, like a chastity belt of sorts. And excuse my crudeness, but it's about the best way that I can explain the attractions that there are there. So it's protecting the actin sites. It's protecting the myosin sites from the actin heads. I've got it all mixed up here. It's protecting the actin sites from the myosin heads. So It's on the actin sites. It's holding onto the actin sites, the tryptomyosin is. And what's holding the tryptomyosin there is troponin. That troponin is holding the tryptomyosin on the actin sites so the myosin heads can't bind to it. But when calcium is present, it causes the troponin to let go of its grip on tryptomyosin. So troponin lets go of the tryptomyosin. The tryptomyosin kind of drops its guard, guarding the actin sites, and the myosin heads make contact and a contraction starts to happen. So I realized that there's a lot of things going on there, but that's how those contractions happen. The energy that it takes for those contractions to happen is provided by ATP. And I know you guys just wrote a paper about ATP and the Krebs cycle and how ATP is created. We'll talk about that more here in a little bit, but the ATP is actually used to set the myosin heads. Basically, it's like setting a mousetrap. It springs them back, and when the actin site is exposed, the myosin head attaches to it and then contracts. And that's how it releases the energy. In order to reset that myosin head, it takes ATP to reset it back in place. So that's kind of what's happening. And I know your book doesn't get real in depth into that, and it doesn't talk about all of those things, but that's what's happening there on a microscopic level when we allow the actin and myosin sites to come into contact with each other. So here it talks about it just a little bit, where the troponin and tryptomyosin play a role there. I've got a video on here. This video actually explains it pretty well of the attraction between actin and myosin and how those work together, and the troponin and tryptomyosin and where those work. Let's watch this video. Hopefully, like I said, once again, if I had the luxury of having each one of you guys sitting in front of me and able to ask me questions so I could clarify this and draw it out on the board, it would be a little bit easier for you. But I'm thinking this video has actually been helpful in the past to some of my students. So let's watch this video. Hopefully it'll clear things up.
Romeo and Juliet, Helen and Paris, Tristan and Isolde. These famous star-crossed lovers bring to mind insatiable longing, forbidden love, and tragic separation, and poets and emo rockers love them for it. But you know where else you could find a nice, hot romance? Your muscle cells. They've got their own famous coupling, a pretty pair of tiny protein strands called actin and myosin. Romeo and Juliet may have set a chain of tragic events into motion with their infatuation, but deep down in the cells of your muscles, the hot protein action between actin and myosin is actually literally causing all of your motions. All of them. And I don't just mean voluntary stuff like walking down the street or moving your mouth so you can talk or chew chips, because your muscles also support your weight and help fend off gravity. The amazing thing about your complicated, self-healing, blood-guzzling muscle tissues is that they turn chemical potential energy into mechanical energy, or movement, simply by doing two things: contracting and relaxing. And that contracting and relaxing is exactly what's fueled by the constant coupling and separation of biology's greatest lovers. Somebody get these proteins a movie contract. You will recall from our early lessons on tissues that you're kept alive and moving by three types of muscle tissue: smooth, cardiac, and skeletal. Your smooth muscle tissue is found in the walls of all your hollow visceral organs like your stomach and airways and blood vessels, where it involuntarily and very usefully pushes fluid and other material around by contracting and relaxing over and over. Your heart is so important that it gets its very own muscle tissue type, cardiac muscle, which looks striped or striated and also functions involuntarily to keep your blood pumping without you having to think about it. But when you hear the word muscle, you're probably thinking of the kind that you see on Chris Evans when he first walks out of that machine in Captain America. And those types, the ones you can see and feel and flex, are your 640 skeletal muscles. They're striated like cardiac muscle tissue, but they're also mostly voluntary, meaning you have to think about using them and activate them with your somatic nervous system. Most of them attach to your skeleton and create movement by pulling bones in different directions as they contract. Each one of your different skeletal muscles, like your biceps brachii, or vastus lateralis, or gluteus maximus is technically its own organ, made up mostly of muscle tissue, but also of connective tissue, blood vessels, and nerve fibers. And because your muscles are voracious energy hogs, each one is rigged up with its own personal nerve to stimulate contraction, and its own artery and vein to keep it well fed with all the blood and oxygen and nutrients it needs to operate. But to understand those operations, we first need to get a grip on the anatomy of a skeletal muscle, which involves fibers within fibers and lots of layers. Basically, a skeletal muscle is constructed like a really sturdy piece of rope. Thousands of tiny parallel threads called myofibrils squished together to form muscle fibers, which are your actual muscle cells, cells with mitochondria, multiple nuclei, and a cellular membrane called a sarcolemma. Those muscle fibers then form larger string-like bundles called fascicles, which combine to form the larger rope-like muscle organ like your biceps brachii. Overall, this bundles of bundles configuration makes muscle tissue fairly sturdy, but considering how much abuse your muscles take when you do something like pretty simple, like lift a big bag of dog food, it's no surprise they need a little help. That's why every muscle contains a few different kinds of supportive sheaths of connective tissue, the protective reinforcements to keep that bulging muscle from bursting. So that is the structure part of the story. But if you want to get into the nitty-gritty, the down and dirty of how you actually move, well, there are rules. Really just two main rules, and they have to do with proteins. And they're both true for a lot of the proteins we talk about, whether they're enzymes or ion channels or receptors or muscle proteins. And these rules are: 1. Proteins like to change shape when stuff binds to them, and 2. Changing shapes can allow proteins to bind or unbind with other stuff. So keep those rules in mind while we see how a muscle fiber contracts and relaxes. Now, remember those tiny myofibrils that bundle up to form your muscle fibers? They're divided lengthwise into segments called sarcomeres, which contain 2 even tinier strands of protein, 2 different kinds of myofilaments called actin and myosin. And it's their angsty story of star-crossed love that fuels every movement your body could possibly dream up. A sarcomere contains both thin filaments made up mostly of 2 light and twisty actin strands, and thick filaments composed of thicker, lumpy-looking myosin strands. Each sarcomere is separated by what's known as a Z-line at either end, which is just a border formed by alternating thin filaments in a kind of zigzag pattern. A muscle contracting is all about sarcomeres contracting, bringing those Z-lines closer together. Alright, so now comes the romance. When your muscle cells are at rest, your actin and myosin strands don't touch, but they really want to. Specifically, that club-headed myosin wants to get all up close and personal with the actin. When this happens, and it will eventually, it's called the sliding filament model of muscle contraction. But in the meantime, like in any good love story, the pair have some obstacles to overcome. Namely, actin is blocked by a couple of protein bodyguards called tropomyosin and troponin, which keep getting in the way. Luckily, these guards can be bought off with a little ATP and some calcium. I'd prefer cash and nachos, but whatever. Remember, ATP is kind of like molecular currency. It contains chemical energy, and your muscles are all about converting chemical energy to motion, so they're always hungry for more ATP. Your muscle cells have lots of nuclei, but some of them also have a lot of mitochondria, whose sole purpose in life is to crank out ATP. And muscle cells also have their own version of an endoplasmic reticulum, the cell's transport and storage system, but in this case it's specialized, so it gets a special name, the sarcoplasmic reticulum. Its walls are loaded with calcium pumps which use ATP to save up a bunch of calcium ions. And it's also studded with calcium channels that are linked to voltage-sensitive proteins in the membrane of the muscle cell. Say I want to move my arm. My brain sends an action potential along the motor neuron until it synapses with a muscle cell in my arm. The receptors on that muscle cell are ligand-gated sodium channels. So when the motor neuron releases our old friend acetylcholine into the synapse, the channels open up and create a rush of sodium into the cell as a graded potential, which, if it's strong enough, causes nearby voltage-gated sodium channels to open. Now I want to take a second and point out here that we're still talking about an action potential, but not in a neuron. This is happening in a muscle cell, people. So that action potential zips along a muscle cell's membrane, the sarcolemma, which has lots of tubes that run deep inside the cell called T-tubules. When the action potential travels down one of those tubes, it eventually triggers the voltage-sensitive proteins that are linked to those calcium channels in the cell's sarcoplasmic reticulum. When those channels are thrown open, the calcium stored inside rushes into the rest of the cell, and finally myosin is like, yes, here we go! At this point, the myosin is totally stoked, because the bodyguards that have been frustrating it are in for a big, irresistible distraction. That's because the protein troponin just loves to bind with calcium, and remember, when stuff binds to proteins, the proteins change shape. So the calcium latches onto the troponin and causes it to pull the other bodyguard protein, the tropomyosin, away from the sites on the actin strands that the myosin really wants to get its paws on. And suddenly it's all, okay. But the only myosin heads that can bind to those newly exposed sites are the ones that are ready for action. That is, the ones that have already grabbed a molecule of ATP that's been floating around and broken it down into ADP and the leftover phosphate. When a myosin head does that, it moves into an extended position, kind of like a stretched spring, still holding on to the ADP and the phosphate, and still storing the energy that was released when they were broken apart. So after all that, with the myosin primed for action and the bodyguards out of the way, the myosin finally binds to actin, and it is beautiful. When they bind, the myosin releases all that stored energy, and in the excitement of it all, the myosin changes shape. It pulls on its precious actin strand, kind of like pulling a rope hand over fist. In the process, it shrinks the whole sarcomere and contracts the muscle. That's the sliding part of the sliding filament model. Now, with its energy spent, that little head has no use for the ADP and the phosphate, so they unbind with it because, remember rule number 2, changing shape encourages proteins to bind or unbind with stuff. That unbinding causes a small change in its shape, which lets a fresh ATP bind there in its place. That binding causes another shape change, but this time it causes the myosin to release from the actin in a tear-jerking scene like some microscopic recreation of the finale from Titanic. But fear not, this epic is not quite over, because this is when the myosin breaks down its new molecule of ATP into ADP and a phosphate, which moves it into the armed position yet again, getting it ready for its next rendezvous. And meanwhile, those calcium pumps are working hard to restock the calcium in the sarcoplasmic reticulum, so they start grabbing the calcium that's floating around, causing calcium to unbind from the troponine. When it unbinds, the resulting shape change puts the tropomyosin bodyguards back into place. It's a circle. Or potentially a big Hollywood franchise with lots and lots of sequels. It keeps repeating itself many, many times every moment while I sit here and talk, and while you sit there and eat and text and take notes, the whole drama replaying itself over and over. Kinda like you'll have to play this video over and over again to get all the little steps of the sliding filament model straightened out. But hey, some stories get better the more you hear them. If you do watch this one again, you will relearn that your smooth cardiac and skeletal muscles create movement by contracting and releasing in a process called the sliding filament model. You'd also relearn that your skeletal muscles are constructed like a rope made of bundles of protein fibers, and that the smallest strands are your actin and myosin myofilaments. It's their use of calcium and ATP that causes the binding and unbinding that makes sarcomeres contract and relax. Special thanks to our headmaster.
All right, so he managed to colorfully explain something that I didn't have a prayer of doing on my own. Hopefully between the repetition of what I've explained and what he's explained in the other videos that we've went through, I'm hoping that you have a pretty good understanding of what it is that we're talking about and how muscles contract, okay? The thing to remember about muscles or a muscle fiber, its contraction philosophy is an all or none. When it gets enough of a stimulation to contract, all of it's going to contract or none of it. So it has to be a strong enough stimulation in order to make it contract. But not all fibers may be stimulated at the same time during the same interval, so it could be more of a graded contraction, which involve different degrees of skeletal muscles. Graded responses have to do with the frequency of the muscle contraction, how often it's contracted, and the number of muscle cells that are being stimulated at one time. So it's a grading. Twitch is just a single brief contraction. It's not a normal muscle function, but we've probably all had one every now and then, or almost like a full body twitch. I'm thinking of the time when you're laying in bed and you're about to doze off and you twitch and you scare yourself awake, or you make your little dog wet itself or something, because they're trying to figure out what in the heck's wrong with you. So that's just a twitch. There is something called a summoning of contractions. It's where one contraction is followed by another, causing more and more frequent muscle contractions, and the muscles don't completely return to a resting state. An example of this is when we go to pick something up and we realize it's heavier and we have to kind of summon more groups of muscles or more muscle groups in order to be able to lift the object that we're trying to lift. We have to go, oh, that's a little heavier than I thought it was going to be. I'm going to have to be a little more serious about this. So the original groups didn't relax, but we've actually added more muscle contracting groups to it. Incomplete tinnitus or unfused tinnitus is when some relaxation occurs between contractions. And complete tinnitus or fused is when there's no relaxation in between. I'm sorry, I'm calling it tinnitus or tetanus, whichever way you want to call it. there is we have to have a tetanus shot, and that's what they're talking about, that the bacteria that if we get exposed to tetanus, it will cause our muscle to go into tinnitus or tetanus and contract and stay contracted, and that's where it gets its name. And that's what they're talking about here. Let's see. The force of that muscle depends on the number of fibers that we stimulate. So the more muscle fibers that are stimulated, the stronger the force that is used. The more fibers, the greater the tension. And muscles can continue to contract as long as they have energy. Think about this when we think about somebody having difficulty breathing and they're breathing really, really fast. They're continually breathing, and they're using those abdominal muscles, and they're using those rib muscles in order to breathe. Compare that to the last time that you had to lift a weight repetitively or lift something repetitively to the point that you got tired and you could no longer lift it. And I'm talking tired to the point that if your life depended on you lifting up that particular object, there was no way that you were going to be able to lift that object. This is what happens when we have somebody that is in what we call respiratory failure, meaning that their lungs are not working properly and they're having to breathe really fast and their muscles are getting tired. Breathing muscles get tired just like any other muscle in the body, and they eventually reach the point to where that they can no longer contract because they don't have enough energy to be able to do it, and they're too fatigued in order to do it. When that happens, we have somebody that's in respiratory failure go into respiratory arrest because the muscles just stop working. Their life depends on them moving air in and out. However, they are no longer capable of doing it because they're completely wore out. This really does happen to respiratory muscles or breathing muscles, just like any other muscle in the body. Okay? ATP. You guys I've read your papers. Your papers were really good. So I'm hoping you have a little bit more of an understanding of ATP, but realize that ATP is that energy source for muscle contractions as well as a lot of other things that go on in the body, right? It's stored in muscle fibers in small amounts, and we use it up pretty quickly. And after that is used up, we have to figure out a way to replace it. As you guys know by now, it takes glucose and oxygen to replace or create more ATP efficiently. We can do it absent oxygen or anaerobically, but it's much better to do it aerobically with oxygen. That is the most efficient way to produce ATP or these bundles of energy that the body's going to need. But there's actually 3 ways to do this. The first one is through direct phosphorylation, and that's where it uses creatine phosphate that's stored within the muscle cells themselves. It can use that creatine phosphate that it's stored. It's a stored version of ATP that the muscles can utilize. Remember, that's going to be short-lived. It's not going to be able to use that for a long period of time. The other one is the best one is through aerobic respiration. This is the one where we take oxygen in our body and it runs the Krebs cycle or the citric acid cycle. And the last one is anaerobic glycolysis and lactic acid formation. We used to just call this anaerobic metabolism, but now we call it anaerobic glycolysis and lactic acid formation because it's important to understand that lactic acid is formed during this process. So not only are we not creating ATP in an efficient manner, we're also creating a buildup of acid within the body. And remember, the body has to stay in that balance. Remember that pH balance of 7.35 to 7.45. Long terms of anaerobic metabolism or anaerobic glycolysis is going to build up a lot of lactic acid, putting our body into an acidic state. And the more acidic that we get, we'll eventually reach that tipping point of damaging proteins to where that they reach the point where they can no longer function. Once we've damaged proteins enough, or once the body becomes acidic enough, the protein hemoglobin protein hemoglobin loses its ability to bind with oxygen. So now we'll have red blood cells that need to carry oxygen because we need oxygen to do aerobic metabolism or aerobic respirations. But now we've damaged the proteins on those red blood cells because of the acidosis, and it's no longer able to carry even what little oxygen that it was able to carry to the tissues. And this problem, can rapidly get out of control and kind of in a catch-22, and we go down the drain pretty quickly. Okay. And this slide just talks about the direct phosphorylation of ATP by creatinine. It's stored in there in the muscle cells, and that's how we do that. And it's just it basically takes ADP, it adds a phosphate group, and it releases creatine and makes ATP. So it just steals the phosphate from the creatine in order to create ATP. It's just a quick way for the body to have a quick reserve. Aerobic metabolism. The great thing about this, if we back up, it's going to take one creatine phosphate molecule to create one ATP molecule. So it's a 1-to-1 ratio. So you got to have a bunch of it if you're going to need a bunch of it. And we're going to need, or that video we just watched showed that we're going to need an ATP molecule to reset that actin head every single time, or myosin head, sorry. We're going to need it to reset that every single time. So it's going to take a bunch. With aerobic respirations, we can create 32 ATP with the use of glucose and water and oxygen. It's a little slower reaction, but it produces a lot of ATP. The downside is it does produce some carbon dioxide. So you've probably known for a very, very long time that your body requires oxygen to survive. We breathe in good air and breathe out bad air. We breathe in oxygen, breathe out carbon dioxide. Well, now we know where all that's going to. That oxygen is actually going into the mitochondria to create ATP, and the carbon dioxide is released from the process of the creation of ATP, and that's what we have to exhale out of our bodies in order for it not to build up, because a buildup of carbon dioxide in the body can actually cause acidosis as well. So that's what's going to happen. All right. Here's a video on the Krebs cycle. It's pretty good actually in how it really breaks down what's happening in there. Pay particular attention to how. Oh shoot, I can't think of the term, the word that I'm looking for. But the very first part of that, before it gets into the citric acid cycle, and what happens if there's not oxygen. And that's a different process. It starts out with the same process, but it's a different process when it splits out. Okay? So here goes. Maybe.
So we already know that if we start off with a glucose molecule, which is a 6-carbon molecule. 6-carbon molecule, that this essentially gets split in half by glycolysis, and we end up with 2 pyruvic acids, or 2 pyruvate molecules. So glycolysis literally splits this in half. It lyses the glucose, and we end up with 2 pyruvates, or pyruvic acids. Pyruvate. And these are 3-carbon molecules. 3-carbon molecules. There's obviously a lot of other stuff going on in the carbons. You saw it in the past, and you could look up their chemical structures on the internet or Wikipedia and see them in detail. But this is kind of the important thing, is that it was lysed, it was cut in half, and this is what happened in glycolysis. Glycolysis. And this happened in the absence of oxygen, or not necessarily. It can happen in the presence or in the absence of oxygen. It doesn't need oxygen. And we got a net payoff of 2 ATPs. 2 ATPs net. And I always say the net there because you remember, it used 2 ATPs in kind of that investment stage, and then it generated 4. So on a net basis, it generated 4, used 2, it gave us 2 ATPs, and it also produced 2 NADHs. 2 NADHs. That's what we got out of glycolysis. And just so you can visualize this a little bit better, let me draw a cell right here. Maybe I'll draw it down here. Let's say I have a cell. That's its outer membrane. Maybe its nucleus. We're dealing with the eukaryotic cell. Doesn't have to be the case. It has its DNA in its chromatin form all spread around like that. And then you have some mitochondria, and there's a reason why people call it the power centers of the cell. We'll look at that in a second. So there's a mitochondria, it has an outer membrane and inner membrane, just like that. I'll do more detail on the structure of a mitochondria maybe later in this video, or maybe I'll do a whole video on them. That's another mitochondria right there. And then all of this fluidic space out here that's between the organelles. Where in the organelles, you can view them as parts of the cell that are, they do specific things. They do specific things, kind of like organs do specific things within our own bodies. So between all of the organelles, you have just this fluidic space. This is just fluid of the cell, and that's called the cytoplasm. Cytoplasm. And that's where glycolysis occurs. So glycolysis occurs in the cytoplasm. Glycolysis. Now, we all know, in the overview video, we know what the next step is. It's the Krebs cycle, or the citric acid cycle. And that actually takes place in the inner membrane, or the inner space of these mitochondria. Let me draw it a little bit bigger. Let me draw mitochondria here. So if this is a mitochondria, it has an outer membrane. It has an inner membrane. If I have just one inner membrane, we call it a crista. If we have many, we call them cristae. These little convoluted inner membrane. Let me give it a label. I'll say they're cristae, plural. And then it has two compartments, right? It's because it's divided by these two membranes. This compartment right here is called the outer compartment. This whole thing right there, that's the outer compartment. And then this inner compartment in here, is called the matrix. The inner compartment right there is called the matrix. Now, you have these pyruvates. They're not quite just ready for the Krebs cycle, but I guess, well, it's a good intro into how do you make them ready for the Krebs cycle. They actually get oxidized, and I'll just focus on one of these pyruvates. And we just have to remember that the pyruvate, that this happens twice for every molecule of glucose. So we have this kind of preparation step for the Krebs cycle. We call that pyruvate oxidation. Pyruvate oxidation. And essentially what it does is it cleaves one of these carbons off of the pyruvate, and so you end up with a 2-carbon compound. You don't have just 2 carbons, but its backbone of carbons is just 2 carbons, called acetyl-CoA. Acetyl-CoA. And if these names are confusing, because, what's acetyl coenzyme A? These are very bizarre. You could do a web search on them, but I'm just going to use the word.
I wanted to stop him here for a second and point out, we can see where glycolysis is happening. It's kind of neat how he broke this down a little bit more inside of the mitochondria, what's going on. Don't get caught up in thinking you need to know what the outer and the cristae and the matrix and all that is. I think it's just good that he's explaining that. But we have, the glycolysis where it splits those 2 pyruvates. We know that there's some ATP and some NADH, which you haven't heard that spoken of before, but it's another source of energy, which he'll talk about that again in a little bit. But glycolysis actually creates these 2 pyruvate molecules. In the presence of oxygen, they'll oxidize. And once they're oxidized, they can actually go into the Krebs cycle then, and we can create more ATP molecules with them. If they don't oxidize, something else is going to happen to them, and they will turn into lactic acid. So I'll let him continue on so he can explain that, but I just wanted to kind of point that out.
Right now, because it'll keep things simple and we'll get the big picture. So it generates acetyl-CoA, which is this 2-carbon compound, and it also reduces some NAD plus to NADH. And this process right here is often given credit, or the Krebs cycle or the citric acid cycle gets credit for this step, but it's really a preparation step for the Krebs cycle. Now, once you have this 2-carbon chain acetyl-CoA right here, you are ready to jump into the Krebs cycle, this long talked about Krebs cycle, and you'll see in a second why it's called a cycle. The acetyl-CoA, and all of this is catalyzed by enzymes, and enzymes are just proteins that bring together the constituent things that need to react in the right way so that they do react. So catalyzed by enzymes, this acetyl-CoA merges with some oxaloacetic acid, very fancy word, but this is a 4-carbon molecule. This is a 4-carbon molecule right here. These two
Guys, no,
guys are
I'm
kind
not
of
expecting you guys
reacted
to
together or
know all
merged
these
together, depending on
names
how you
and
want to do
remember
it.
all those, so don't worry about
Draw it
that.
like that. It's all catalyzed by enzymes. It's important, some tests will say, is this an enzyme catalyzed reaction? Yes, everything in the Krebs cycle is an enzyme catalyzed reaction, and they form citrate or citric acid. Citric acid, which is the same stuff in your lemonade or your orange juice. And this is a 6-carbon molecule, which makes sense. You have a 2-carbon and a 4-carbon. You get a 6-carbon molecule. And then the citric acid is then oxidized over a bunch of steps. And this is a huge simplification here, but it's just oxidized over a bunch of steps. Again, the carbons are cleaved off. Both 2 carbons are cleaved off of it to get back to oxaloacetic acid. And you might be saying, hey, when these carbons are cleaved off, like, when, let's say, this carbon is cleaved off, what happens to it? It becomes CO2. It gets put onto some oxygen and leaves the system. So this is where the oxygen or the carbons or the carbon dioxide actually gets formed. And similarly, when these carbons get cleaved off, it forms CO2. And actually, for every molecule of glucose, you have 6 carbons. When you do this whole process once, you're generating 3 molecules of carbon dioxide, but you're going to do it twice. You're going to have 6 carbon dioxides produced, which accounts for all of the carbons, right? You get rid of 3 carbons for every turn of this. Well, 2 for every turn, but really for the steps after glycolysis, you get rid of 3 carbons, but you're going to do it for each of the pyruvates. So you're going to get rid of all 6 carbons, which will have to exhale eventually. But this cycle, it doesn't just generate carbons. The whole idea is to generate NADHs and FADH2s and ATPs. So, right here, and this is a huge simplification. I'll show you the detailed picture in a second. We'll reduce some NAD into NADH, NAD plus into NADH. We'll do it again. And of course, these are in separate steps. There's intermediate compounds. I'll show you those in a second. NAD plus, another NAD plus molecule will be reduced to NADH. Maybe we'll produce some ATP will turn into, or some ADP will turn into ATP. ADP turning into ATP. Maybe we have some, and not maybe, this is what happens. Some FAD gets, let me write it this way. Some FAD gets oxidized into FADH2. And the whole reason why we even pay attention to these, you might think, hey, cellular respiration is all about ATP. Why do we even pay attention to these NADHs and these FADH2s that get produced as part of the process? The reason why we care is that these are the inputs into the electron transport chain. These get oxidized, or they lose their hydrogens in the electron transport chain, and that's where the bulk of the ATP is actually produced. And then maybe we'll have another NAD get reduced or gain in hydrogen, right? Reduction is gaining an electron or gaining a hydrogen whose electron you can hog. NADH. And then we end up back at oxaloacetic acid, and we can perform the whole citric acid cycle over again. So now that we've written it all out, let's account for what we have. So depending on, let me draw some dividing lines so we know what's what. So this right here, everything to the left of that line right there is glycolysis. Glycolysis, we learned that already. And then most, especially introductory textbooks, will give the Krebs cycle credit for this pyruvate oxidation, but that's really a preparatory stage. The Krebs cycle is really formally this part where you start with acetyl-CoA, you merge it with oxaloacetic acid, and then you go and you form citric acid, and which essentially gets oxidized and produces all of these things that will need to either directly produce ATP or you do it indirectly in the electron transport chain. But let's account for everything that we have. Let's account for everything that we have so far. We already accounted for the glycolysis right there, 2 net ATPs, 2 NADHs. Now, in the citric acid cycle, or in the Krebs cycle, well, first we have our pyruvate oxidation that produced 1 NADH. But remember, if we want to say what are we producing for every glucose, this is what we produced for each of the pyruvates, right? This was from 1 NADH from just this pyruvate. But glycolysis produced 2 pyruvates. So everything after this, we're going to multiply by 2 for every molecule of glucose. So I'll say so the pyruvate oxidation times 2 means that we got 2 NADHs, 2 NADHs right there. And then when we look at this side, the formal Krebs cycle, what do we get? We have how many NADHs? 1, 2, 3 NADHs. So 3 NADHs times 2, because we're going to perform this cycle for each of the pyruvates produced from glycolysis. So that gives us 6 NADHs. We have 1 ATP per turn of the cycle. This is going to happen twice, once for each pyruvic acid. So we get 2 ATPs. And then we have 1 FADH2, but we're going to do this cycle twice. This is per cycle, so times 2. We have 2 FADHs. Now, sometimes in a lot of books, these 2 NADHs, or per turn of the Krebs cycle, or per pyruvate, this 1 NADH, they'll give credit to the Krebs cycle for that. So sometimes instead of having this intermediate step, they'll just write 4 NADHs right here, and you're going to do it twice, once for each pyruvate. So they'll say 8 NADHs get produced from the Krebs cycle. But the reality is 6 from the Krebs cycle, 2 from the preparatory stage. Now, the interesting thing is we can now count whether we get to the 38 ATPs promised by cellular respiration. We've directly already produced, for every molecule of glucose, 2 ATPs and then 2 more ATPs. So we have 4 ATPs. 4 ATPs. How many NADHs do we have? 2, 4, and then 4 plus 6 is 10. We have 10 NADHs. And then we have 2 FADHs. 2 FADH2s. I think in the first video on cellular respiration I said FADH. It should be FADH2 just to be particular about things. And these, so you might say, hey, where's our 38 ATPs? We only have 4 ATPs right now. But these are actually the inputs in the electron transport chain. These molecules right here get oxidized in the electron transport chain. Every NADH in the electron transport chain produces 3 ATPs. So these 10 NADHs are going to produce 30 ATPs in the electron transport chain. And each FADH2, when it gets oxidized and gets turned back into FAD in the electron transport chain, will produce 2 ATPs. So 2 of them are going to produce 4 ATPs in the electron transport chain. So we now see we get 4 from just what we've done so far, glycolysis, the preparatory stage, and the Krebs or citric acid cycle. And then eventually these outputs from glycolysis and the citric acid cycle, when they get into the electron transport chain, are going to produce another 34. So 34 plus 4, it does get us to the promised 38 ATP that you would expect in a super efficient cell. This is kind of the theoretical maximum. In most cells, they really don't get quite there, but this is the good number to know if you're going to take the AP Bio test or in most introductory biology courses. There's one other point I want to make here. Everything we've talked about so far, this is carbohydrate metabolism, or sugar catabolism, we could call it. We're breaking down sugars to produce ATP. Glucose was our starting point. But animals, including us, we can catabolize other things. We can catabolize proteins. We can catabolize fats. I mean, if you have any fat on your body, you have energy. You're in theory, your body should be able to take that fat and you should be able to do things with that. You should be able to generate ATP. And the interesting thing, the reason why I bring it up here is obviously glycolysis is of no use to these things, although fats can be turned into glucose in the liver. But what the interesting thing is that the Krebs cycle is the entry point for these other catabolic mechanisms. Proteins can be broken down into amino acids, which can be broken down into acetyl-CoA. Fats can be turned into glucose, which actually could then go the whole cellular respiration. But the big picture here is acetyl-CoA is the general catabolic kind of intermediary that can then enter the Krebs cycle and generate ATP regardless of whether our fuel is carbohydrates, sugars, proteins, or fats. Now, we have a good sense of how everything works out right now, I think. Now I'm going to show you kind of a diagram that you might see in your biology textbook, or I'll actually show you the actual diagram from Wikipedia. And I just want to show you this looks very daunting and very confusing, and I think that's why many of us have trouble with cellular respiration initially, because there's just so much information. It's hard to process what's important. But I want to just highlight the important steps here, just so you see it's the same thing that we talked
On your test, you're going to have a diagram that you'll have to label all that. No, I'm kidding. You won't have to do that. I want you to have just a really good surface understanding of the Krebs cycle and that kind of thing. And as you move on in your studies and you dig deeper and you take higher level classes and those types of things, you'll get deeper into it. But I really wanted to help create a solid foundation. And that's why I opted to go a little bit deeper than what your book's going. I know there's one looming question that all of you probably have. You're probably thumbing through your book right now or looking somewhere online. So get your attention back to me here for just a second. When Saul talked about this, he talked about how this Krebs cycle created 38 ATPs. And your book says 32. I'm pretty sure that's what your book says. Well, remember, he was talking about a hyper efficient cell creating 38 ATP. Skeletal muscle cells generally create somewhere around 32 to 34. If there's any tests about the number that they create, or if there's any test questions about the number that they create on my test, that will reflect what your book says, which I believe is 32. So, but be aware that not every single cell is going to create the exact same number, but it's going to be in the 30s. I don't think there's any that create any more than that. So. All right? So that whole process is important. And let me just back up because I'm pretty sure it'll just tell us. Yeah, 32 is what your book says, is about 32. Some do create more than that. So remember, the process takes about 2 to get things started. It's going to break that pyruvic acid. It's going to break that glycolysis is going to break that glucose into 2 pyruvates. If there is no oxygen, that pyruvic acid will actually be converted into lactic acid. If we don't use it, if we don't oxygenate it to create the acetyl-CoA to start the Krebs cycle and then the electron transport chain to create all that ATP, if we don't use it, it's going to be converted into lactic acid and it's going to be stored and it's going to be in the bloodstream and all that stuff and create problems. This aerobic version of this, absent oxygen, only goes as far as glycolysis. It's just glycolysis is all that it is. Remember, glycolysis is the first step in the Krebs cycle. But absent oxygen, it never gets to go any further. So all we get is glycolysis. And it's horribly inefficient in what it can do. It creates 2 ATP molecules, but remember, it created 2 NADH molecules as well. But if those NADH molecules never get a chance to get to the electron transport chain, which is the last part of that Krebs cycle, if they never get there, they never get converted. So they never get used. They would actually create how many did he say, 4 to 1 or something like that? They would actually create quite a bit of ATP. However, it's never going to get there because we don't ever get the Krebs cycle kicked off without oxygen. A very, very poor way to create ATP, and it's horribly inefficient. We talked a little bit about this. We're coming to the end, so not going to cram much more into your brains. So bear with me. Muscles do get tired, and they do run out of oxygen. The more we work them, the more strenuous and prolonged the activity is, the more ionic imbalances occur, the depletion of calcium and sodium and potassium and that kind of stuff. Lactic acid's going to accumulate in those muscles because what's happening is once we reach the point where we're getting into an oxygen deficit at the muscle cells, it's going to start creating lactic acid. It just does that. Unless you're just a super efficient, superly physically fit, it's going to do that. And eventually ATP supplies are going to decrease. We're not going to be creating as much. We're going to be building up lactic acid. Muscles are going to get tired. They're going to get sore, and they're going to stop working. So that's what happens as we progress through exercise. And I pointed out that that's exactly the same thing that happens if we have somebody that is, say, in respiratory failure and their lungs aren't working right, but they're trying to breathe more air in and out, but it's just not working effectively. Those muscles that are moving that air in and out of our body will get tired, and they're not going to keep up, and they'll quit. This goes in and talks about isotonic and isometric contractions, and it has to do with the tension and the sliding filaments and the way that they shorten and that kind of thing. I'm not really going to test you on those 2 theories. I just wanted to leave them in there, so I didn't want to just completely skip them over. But isotonic is kind of like the bending of the knee or rotating of the arm. But an isometric is when you're like pushing against something with your elbows bent, like you're trying to push a car or something like that. It's two different types of examples. Setting up, standing up, me wandering around the classroom, I need to have good muscle tone so that my muscles are ready to react. It also helps me stay upright and stand, part of that muscle tone. But the problem is if we destroy the nerve supply to that muscle's no longer going to be functional. It's no longer going to be useful. The muscle will begin to wither, shrink, die, all that kind of stuff because it can't move. It doesn't have a nerve impulse going to it anymore. And if we're not using it, we lose it. Remember, form follows function. If it's no longer functional and no longer usable, it's not going to look normal. If we see somebody that's paralyzed, say from the waist down, after they've been paralyzed for several months or a few months anyway, we'll see that their legs begin to get real thin and frail and small looking. Even their bones become more porous because those parts of the body are not being stressed because they're not being used. And the function of them has changed. So they're basically just limp limbs laying there that are doing nothing. So the body's not going to expend any kind of energy and things like that to preserve them for the state that they once were. So that's how it's going to work. Exercise. When we exercise, it does help our muscles. It builds up endurance. It actually makes our muscles stronger, a little more flexible and that kind of thing. It even helps us build up our stores, our oxygen reserves in our muscles. I talked to you about, I've mentioned hemoglobin. I haven't talked about specifically what hemoglobin does and how it works, but hemoglobin is on red blood cells, and that's what carries oxygen to the tissues, right? Well, the more we exercise and build up muscles, the more we stress our muscles to the point where they go, hey, look, we need to be ready for anything. Muscles have the ability they have a specialized protein within them as well, and they can mass produce the specialized protein called myoglobin, which is very similar to hemoglobin, that they can store oxygen reserves on. There's also a similar protein in the brain. The brain has an ability to store some oxygen reserves there as well. But that's a way that it can store, and this is in part how when we stress our bodies and we try to get in shape, as we call it, this is what happens is our body reacts to that and creates ways that it can respond to those stressors that we're putting it under regularly, and it builds up endurance and exercise and those types of things. All right? So guys, that's chapter 6. I realize there's a lot of information to it. Review those videos, seek out some other information, send me an email. I don't mind visiting with you if you're struggling trying to understand it. I know I threw some additional information in here that's not necessarily in your book. Hopefully it helps you understand it a little bit deeper than what your book was going, and I'm not doing that to pick on you. And no, I'm not going to put really tough, hard test questions in there. But the reason why I've opted to do this is I opted to keep that stuff in there, or to put that stuff in there to deepen your knowledge of it and hopefully make it easier to understand what I will be testing you on. I hope that makes sense. Please just trust me and don't hesitate to send me an email with some questions, and I'll be glad to clear it up as best I can. See you at chapter 7.